What's up, everybody? Welcome back to a new video. This is a new series I'm going to be doing. You guys know I'm doing my PhD, so I have a lot of content that to read on during my research. So I just thought, why not just share it with you guys? So one of the things that we have to do uh, every few weeks, every week, is to basically read a new journal and get to know about the newest development in the research field. So I thought it would be very interesting and really fun to actually break down some of these papers that I've read into simpler terms for you guys to understand. So whether you guys have any other feedback or anything of the video, and if you like what you're watching, you got a like or even follow, or you can watch me live as I play other games as well throughout the weekend. So without further ado, just jump straight into this. So welcome to a new paper. This paper is about RNA codon expansion via programmable pseudo-uridine editing and decoding. It was by a China group. You guys see the names of people and authors that are people involved in this project. Credits to them for being able to publish this paper and doing the work. So next, I know there are many big words here. RNA codon expansion via programmable pseudo-uridine editing and decoding. But let me break it down for you guys. Right, first, some quick background on the expansion of genetic code. So what does it mean by expansion of genetic code? So it's not indicated here. Let me just grab my pen and let's just talk about it. So generally, when we are talking about any form of protein or genetic expression, we're looking at a series that goes like that. In your body, you have proteins are what determines the function. So how do you get to the proteins? First, we have what we call your DNA, right? Right here. I don't think, sorry, I don't think you guys can see this. Right. So you have your, you can see here, the DNA. So this is what determines of how you look like, what kind of blood you have, what kind of organs you have, your skin color, the eye color, all that. Your genes, essentially, your DNA. Then the next, after DNA doesn't go straight to a protein, they go through an intermediary, what we call RNA. It's like a copy of your DNA, but it's slightly made up, slightly different. And your body requires this RNA to be read to then be able to be expressed in the protein, right? So how do the RNA be read into the protein? Generally, these are determined by a triplet codon, what we call 3 letters mixed together. So generally, for example, for DNA, it's around A, T, E, and G. And RNA is very similar, except it's a A, U, and T. So if you have a series of letters, every 3 sets of letters will determine one kind of amino acid that is going to be determined. Right? So what we call by genetic code is just expand. It's such that we made it such that this triplet codon can code more than what is able to code initially, right? So if you do a quick math, essentially if you have 4 different kind of codons, there's some redundancy involved. In total, you will be able to 64 codons will be able to code for around 24 amino acids, if I remember correctly. So however, there are some repeats. For example, we talked about a stop codon. The stop codons are what stops a protein from being read through the RNA. So one of the things people do is actually to reassign stop codons. There are 3 different kinds of codons, 3 different kinds of sequences that determine a stop codon. A bit redundant, as you can see. So people make the extra ones to code for other kind of amino acids that is not natural in the body. Okay. So here you can see, right here in this image, you see what generally is how it's being made. So first, it goes from a amino acid. If a tRNA will be go through a reaction called amino acylation. Not very important, it's just essentially that's just using an amino acid to a tRNA. And this tRNA has an anticodon, which is again the 3 letters that is able to recognize the 3 letters on your RNA. And by recognizing, you're putting it to the right spot, and this amino acid is what makes up a protein. would then be linked together to form your protein eventually with different function. Right. Essentially that's what gene expression is. Expression is actually now your codon can code for more things. So apply it for differently, we people do it in imaging. So you can have long amino acids that don't have some kind of chlorophyll and all that being fused to it. It's able to control protein function. You're able to use different kind of chemicals that prevent protein functions to occur, and only by changing the chemical that's being fused, the function can come back. And this you can actually look into discovering or exploring what the protein function is involved in and how it changes throughout the body. Right. And lastly, people like to do what people like do is called therapeutic strategies. What I mean by that is they exploit these to make medicine. One of the things that people do essentially uh, is, for example, in here is that they introduce what we call these other stop codons into genomes of this Hep B virus, HIV, or even influenza A. And this is such that only when these viruses can only replicate in cells that contain these amber codons. So this, in the sense, would actually help the production of various viruses for your what we call your antibodies that you take, your influenza antibodies and all that you take. The vaccines, yeah, the production of the vaccines. Okay. Next, so one of the problems that people face when they assign, so typically when people talk about reassigning stop codons are involved in DNA. So one of the problems that people have is off targets. Because if you reassign or stop codon to something else now, these proteins now may not combine properly anymore because both a stop codon and the non-canonical amino acid cannot bind to it. And of course, there are other strategies that are involved to change this, but typically these are not suitable for mammalian cells, which are the human cells. Things that people have done is, for example, instead of 3 amino acids, they recognize to make it a quadruplet codon, or they actually make a genomically recoded organism. They make codons code for something else totally different again. So, for example, you have what we call a Sim61 E. coli, which is a bacterial cell, which now only codes of 20 different chromatic amino acids using their 59 different sense codons. Right. This is not possible a lot of times in mammalian cells due to its toxicity and complexity of nucleotic human. So now people think is that if we cannot make DNA stop codons, what about if we make RNA to stop codons? And this could be possible because RNA is beyond just the letters, you can actually modify the RNA. So there are so many different RNA modifications, up to 150 of them, and they have effects independent of DNA codon. It's perfectly possible to create an RNA blank codon outside of the 64 genetic code that commonly essentially the 64 codon code for that. So the overall strategy of this paper that they managed to discover is split into mainly two parts. Number one is the production of pseudo-uridine codons on the intended mRNA and intended target. Second is to decode this pseudo-uridine codon. Oh, I forgot to talk about it. Pseudo-uridine is just a modification of the uracil or U that you see in your RNA. They add a chemical compound to it. So you can see it changes from carbon here to an amino group. That's about it. Simple as that. So I think the interesting part is kind of decoding when it forms tRNAs that actually can bind to these pseudo-uridine codons first right? So number one strategy, how do they install pseudo-uridines on specified mRNAs? We did not make this. This has been discovered previously. People have found this, which is called MeSTAR. So it's what we call RNA editing to a specific transcript of pseudo-uridine mediated premature termination codon with 2. So what exactly is this? We just have what we call an enzyme guide RNA, and we with the engineered guide RNA, and we also have we make use of the endogenous system in the body. It's the casing box, which actually does this within the action. And you do this to you don't reliate the target that you have. It's CRISPR free. The CRISPR is something totally that I'll not touch about here today, but it's actually just a bacterial system that we use to actually modify nucleic acids. It's a state of art that's what we're use right now. something interesting about restart, but they wanted to test to make sure it's great. So first, they started with UGA subcodons. So they're one of the most prevalent subcodons in the body. So like 52% of this of UGA or subcodons are UGA. Right. So the first, we just focus on here first. So they first of all they installed a stop codon within this area and these targets, right? And then it's linked by it's in between these GFP reporter and cherry linker GFP reporter. So next, they also want to see how much the UGA has been installed. So they use trace, which is a method that is able to find out how much of this pseudoresidue is actually added into these sites. This is done through sequencing, bisulfite sequencing, which will actually result in the deletion in the area where you install the pseudoresidue. So by seeing how much deletion, you know how much pseudoresidue is observed. So if you focus first on these figures, B and C, right here, right? B and C, you can see that most of this is the pseudoresidue is highly installed into these zones. Pretty much there's not much differences. Different sites that pretty much is all the same, right? And then we tried to test the different stoichiometry, so different amount of these RNA that they actually added. And then you can see that pretty much the installation efficiency is still very, very high. Okay. Next, they actually want to test what if you change the amount of tRNA that is added, and then you can see that it doesn't really affect the COD ratio. So this can be seen pretty much all the same, so 75% of the ratio of COD addition, right? Next, so they wanted to look at off targets because now it's a very important thing. Whenever you want to target something like nucleic acid, you want to make sure it does not target anything else, right? So you can see that the ratio is relatively low. You can see, well, yeah, exactly. So you can see the off target is relatively low in this system, pretty much non-existent. Yeah. Okay. So it's kind of expected what they see, because generally most of the time when this off target, you can see that it's the motifs that actually resemble the on target sequence. If you look here, you see the off target sequence, yes, two more amino acid lesser than it binds to, so that results in the off target that you see. And then yeah, you can see it in off target, you can see here it all resembles pretty much the same as what we have in on target, even the motifs as well. And then in low, you can see that if you look at here, if you look at the off target sites, you can see generally these are not really any endogenous stop codon, as well as there's a very low effect of the RNA expression of the somatostatin. So this is great. So means whatever that they did to the CDD had little impact. Next, so if you stop pseudouridine, you need some way for this tRNA to be assigned specifically to pseudouridine and not impact the normal UGA that we have. You need something to beat pseudouridine specifically. It's very important because actually UGA, as I mentioned just now, is 2% of all stop codon. And it's actually very easy to be read through, means it can be bypassed. So you can imagine if you have the tRNA not specific, it's going to be there's so much off target. There's so many kind of off proteins that cannot go properly because the protein itself is not done properly. So how they first started is they look at 3 different types of tRNA, which shows minimal ritual differences. So 3 of them that are identified right here, the 3 different ones, the 3 different species. They particularly only focus on the engineering on the anti-codon stem, and the reason is because this ASL and this stone loop, anti-codon stem loop, has equal stability, flexibility, and proximal interactions of the tRNA to the target anti-codon site. Right. So they also only want to focus on ASL, which because it does not affect the direction of the code of the enzyme that is involved in the amino acylation in the tRNA, and you don't want it to affect so that this uh, enzyme can still work on the tRNA variants, can still generate these variants without affecting its amino acylation activity. Yeah. So again, same reporter, GFPN cherry here with a premature codon inside. So actually what they see is that you'll see if you N cherry is produced, you'll see N cherry. However, if the stop codon is big, you can read through, then you'll see both colors on in the cell, right? So based on this, you can read here. So it's open. Oh, right. So they essentially at these sites they mutated every single nucleic acid to every single type of nucleic acid on every spot at this region. Most of them generated 1.5-fold, at least 1.5-fold power mutation, 1.5-fold history within GA preference. And the January 27 is a show results in the highest amount of preference. So this is what you kind of expect. So you see this. So smaller means more specific, higher means better. So this one is you can see it's very high, 0.3 be true only a single theory reading and only 0.1 lower. This is only like 10% history. And it's really background. This is something that we want. Right. So this is what we have. So in the end they selected this 3017 mutation. Okay. So they pick this, they want to see if it's really that good. Right. So they chose on 3 different kind of genes to make sure it's robust, so it's not ticket dependent. Uh, so they chose 200 fragments in the center. You can see right here, they put a fragment reading here, which they can change. And you can see that essentially these proteins are all these theories all relatively high in this different site. Well, there's one at almost 800% to 2%, and around 2%, so say lower than 2%. And you can see with all these different kind of fields, you can see that there's very high preference for this pseudo unity. You can see when they add a non-canonical amino acid, it is at K. It is at K, you can see IDC and RC is already very high. It's very much preferred on these sites. If UGA is typically low, you don't see any addition of these sites. UGA, you cannot find any of that. And even on if you look at the cell culture, you can see that only the presence of the non-canonical amino acid, the tRNA, then can you see the GFP come back? Right. It's something that which is good. Except this maybe the XGT is a bit more unspecific, right? And so you see a bit of GFP coming. You know, but if it is the case, it's still very specific. Next, they want to validate even more, so they do in 4 different cell lines as well, and they see the same result. Okay, so next they want to confirm that these are tRNA actually decode pseudouridine specifically. The non-connected amino acid and like natural amino acids like amino in the body don't just bind to the pseudouridine as well. So they done a liquid chromatography mass spectroscopy and liquid chromatography tandem mass spectroscopy and the tRNA charging with acid using gel northern blocks to look at that to see this. So here is just a schematic workflow how the experiment is being conducted. We have the proteins, isolate it, digest, and then do LCMS or MS, and then we can identify the kind of amino which are being present in the site. And you can see that in the presence of pseudouridine, you can find that only the non-canonical amino acids can be found here, and all other amino acids are generally not found in these regions. Right? Next, we also can see in using the acid raging northern blots, we can see only the presence of the non-canonical amino acids is being added. Besides these are being added, you can see it's much larger, which means is being added. So it's very much specific of design. Right? And then, so they want to also check, so this is a guy with a stress response. So they wanted to make sure that any of these funny mutations and all these occurrence does not cause stress to be activated within the system. And when they check at that, you can see the translation level of these stress related genes are generally not increased, they still generate the same. So all good dedicated as well. None of these is changed. If you look at the Western blot, nothing is particularly upregulated as well as stress. So this is as not really realistic. So they use a tumescimycin as a control because it's supposed to upregulate stress not stress increase. So that the structural basis of why that mutation works. So essentially, bottom line of this, the reason why this RNA is specifically more specific to pseudo-UBB is because this increase in hydrogen bonding towards this region. They have 3 more hydrogen bonds as compared to the yeah. So then so we talk about the coding specificity of this RC system. They want to check how specific this is. Every time when we talk about nucleic acid changes or genome engineering specificity, off target is the most important thing for. So of course they want to see the sustainability actually binds to other products, right? So they look at UAA and UAG, you can see no change. Pretty much is the same. It does not actually particularly bind to these regions. There's no optical regions at these regions. And then for on targets, you can see article is similar in both the PCE system and the RCC system, which means they're both on color in terms of on target. However, you can see in terms of the off target, you can see the RC is quite much lower as compared to the on target. So it's like 0.057 on target and 0.01 point off target for us. So there's almost a 4-fold reduction in terms of the off target, which shows the specificity of RC system compared to PC, the genomic expression system. Again, this illustrates how the RNA codon expression system is more specific than genomic codon expression system. We look at it specifically. It's actually the same experiment. However, what they do is that they set an R value as a cutoff for off target. Anything that's above this value is high, and regardless of what kind of value that they put as a cutoff for the whole change, the R system is always more specific. And even more importantly, when you look into what kind of genes are being off targeted here, right? Off target is only enriched in four pathways for RC compared to GCE. It's so highly affected, and a lot of things are really what we call DNA damage. So they're very enrichment of DNA damage and repair for RC system. Generally, they all revolves around RNA ribosome biogenesis. And then again, typically when you talk about read-throughs, typically when you talk about read-throughs again is when the codon is being bypassed, stop codon read-throughs. It's involved in a lot of diseases. So they want to see if you can see this kind of naturally occurring read-through events in usually codon. Okay, I've got 2 different genes. So one is the MDH1 and the other is THGL1. The properly distinct read-throughs in control cell. And when you look at RCE cells, you can see that the RCE read-through is pretty much almost the same. It's slightly elevated, but pretty much the same as control. But GC is significantly enhanced. This can be seen in both the MDH1 and also in THGL1. Right. And for RCE, you can see there's no clear off-target read-through. and there's no interference of the transcriptome in either the RC or DC cell. So this indicates that interference of the codon expression methods probably occur in the transcriptome, but not in the transcriptome, considered as the previous report. So kind of expected because you're talking about amino acids, we're playing around with translation. Next, so now we come into the application of these various non-codonic acids. First, okay, before that, you want to see how site specific these non-codonic acid coding by us, how specific these non-codonic acids are being added. Look at, they use 4 different kind of non-codonic acids that represent different kind of things they can do. Some is labeling, some is investigating protein function. Right. So first they selected a protein to investigate its SRC kinase. It's a well-known non-receptor protein tyrosine kinase that participates in many of the signaling pathways. So always the method of signaling pathway is cascade of phosphorylation. So tyrosine kinase is involved in phosphorylation, and in particular, lysine 295, which is site on the protein, is crucial for this phosphorylation. So they made a 295 mutated construct, so this is non-functional. And only when a non-canonical amino acid is incorporated within this residue site can the protein be activated and be normal again. Or it just go in this region so that you can control the protein function. They also made a mutation of Y527F, which abolishes onboarding inhibition. So your NC will forever be phosphorylated, like continuously phosphorylation. Incorporate 4 different amino acids, 4 different types. You can see that these something I say pretty much very specific to these features be true. But doesn't really bind to UGA or specifically only to the secondary portion. And it incorporates as well as the what we call cumulative codon expression. The trend now is application, so they want to see you can do this. So they chemically cage and non-connected amino acids undergo bio-orthogonal cleavage reactions. So it's generally just you put in another non-connected amino acid in the presence of another chemical, it will be cleaved and the process can function. That's about it. So they use the TCOK or PCOP to do this. So now PCOP will be within, again, just remember I'm about 295, right? PCOP is your 295, and the non-connected amino acid there cannot function. Only when you teach the function will come back. So you can see number 1, again, if you look at SRC, pretty much the amount of SRC is the same. You can see high level of SRC is easily seen. The protein is fully functional, protein expressed. Next, you can see very high amount of phosphorylation as well. You are down, it's one of the sites that is being phosphorylated. And then lastly, we can look at this Y-positive phosphorylation. This is a target site for SRC, and you can see it's highly phosphorylated only when this protein is depleted. So this tells that this is functional, performed as expected. We talk about phosphoactivation. Same thing again. They wanted to see if there's any. When you look at all the phosphotyrosol sites, you can see it's clearly upregulated whenever you add in the repeat thinking. It shows that when you deplete, you can couple the caging and the function together. And then they all have very similar motifs as the SRC, right? You can see function. Yeah. So a lot of them is overlapping what you can see. You see lah, essentially. In how specific, but I think it can function application of this non-connected amino acid is the specific is being able to label protein. We are label the proteins. So you can see here they add in a tag book which actually do chemistry to bind to a protein. Then you can see then you can be bioaccumulated and then you can pull down and all that nonsense and then to You see here it's just showing that only when taboo that is specific to taboo, in taboo high incorporation, but this is only detected when you have taboo inserted, and then it's much more specific and competitive. That's all I'm trying to say. And then into the off target, again, we look at off target, you can see that we don't target anything else. And then you can see for GC, there's so much high biosignal intensive and non-target GC. So again, it illustrates how non-specific GCE is, but RC is so much more specific. It's almost the same as the top. So, and any off targets incorporation of RC is merely a subset of what you see in GC. So again, it shows how specific the RC is. Now, I just want to talk about subcodon U shape A, right? With a more two other subcodons, which are UAA and UAT, and try to do the same expansion of mRNA codons on them as well. And they found using the same process, they're able to, first they can incorporate this pseudo-UGA over up to 100% in this country. If not, and do a similar screening process to find the tRNA as CGA. They first identified a clear area which shows specificity to this. It's an mRNA mixed RNA. Again, it's 37 gene mutation as well. You can see it's highly specific towards the pseudo-UGA codon. For UAG, it's slightly different because a single mutation is insufficient. We need two mutations. I identified 31G, 37G as the most ideal one. It's much. It has 2.14 UAG preference and 15% H3 efficiency. And thus is selected for it. So it's 15% compared to other one under 10%. It's very low background. Then it further testing with UAG, and you can see that you can use against this UAG system to actually control how the protein is being localized. Only when it's using that's using AG, only when the protein is being active inside AG, and then it can protein be localized properly into the liquid. It's all the endoplasm into cytoplasm, and now it's not all inside the and again, and yeah, it's highly recruit for this specific. You can see that all these various tRNAs with the new tRNAs that we have discovered or mutated is only specific towards the codon that they are specifically testing. Next, they also want to show that you can actually do both at the same time with careful engineering. So they use RCUG system paired with a GCUAG system to incorporate two different kind of non-connected amino acids. So first, SRC catalytic ability relies on lysine 295, which is inhibited by Y527, right? So they put a T-COP residue at 295 and a residue at another SAMP 527. So they managed to use one tRNA first to incorporate into the GC system for the B-COP, and then for RC, they managed to install T-COP inside. They make sure it doesn't affect each other. And then they can still, and this tRNA is still can still observe strong UGA, UGA preference, and doesn't actually affect the UGA normal one in the GC, even though it's present. So engineering can still make sure that both systems can work together and still together. So in conclusion, this RC strategy is highly specific and highly efficient. It's independent of silicon photons. There are 150 kind of modifications that play around there and really like go nuts about what kind of photons you can have. It can concur with other editing strategies because in the sense of you can install different kind of modifications in many parts of the mRNA ideally, and it also can be adapted to other. That comes to the end of the paper. I hope you guys learn something from this super long thing. Let me know if you guys have any feedback in the comments or anything. Let me know what you guys like to learn more about as well. So I hope you guys have a good time here. So catch you guys live whenever I go live. So peace.